Right. So, um, today we're going to look at, we're going to finish up lecture 8, which was exploring the cerebrum and higher order functions. There was a few knots that weren't tied up at the end of this, and then we'll go into the CSF and reflex lecture, which is pretty short, so it shouldn't be too bad. Um, So remember in lecture 8, we saw the forebrain was in two parts, the diencephalon and the cerebrum, right? The telencephalon. We talked about communication in the CNS via pools and tracts, right? We saw such as the corpus callosum, how the two hemispheres talked. We saw a bunch of that. We talked about functional groupings, the limbic system being those emotional areas, the reticular formation, was more of those arousal and alertness areas. And last but not least, what we need to cover is just some questions about complex functions generated by the brain. And to be fair, we don't give it justice in this course, but these things are still being discovered and they're a little bit gray zone of our understanding. But hopefully, over the course of the nervous system, you kind of realized how certain things humans do are a little bit more robotic than you might have thought they were, right? Although we do them so smoothly and we move so smoothly and with not choppy, right? At the same time, there's these, there's this pattern in back of it that allows us to do that, right? So I'm going to take a look at some complex functions. We're going to look at 4 questions. So first of all, how are complex behaviors like memories and emotional states communicated across the brain? And for that, we're going to need to take a look at some multimodal association areas. I think we talked about it last time. That includes the prefrontal cortex. Um, what exactly is memory and how do structures of the brain create memories, right? For that, we need to kind of define what memory is and consider what's important in its creation. We're going to take a look at that. Very interesting topic. How do scientists monitor brain activity? This is using an EEG. We'll talk about the electroencephalogram, um, and this is often used even in hospitals, so you'll be um, accustomed to this tool eventually. Um, why are some people considered left or right brain dominant, and what does that mean? We'll talk about lateralization and how our cerebrum is, there's a dominance pattern with one of the hemispheres being the dominant one. We'll talk about that. So, how do we perform complex actions, right? We talked about these primary areas, association areas, but now we need an area where everything converges, all the information, right? And these are called multimodal association areas. Multi means many, and modes means like the different modes, the different senses that we have: vision, hearing, smell, all of these multiple of those senses, right, associate in certain areas. And we talked about all these association areas, but they were just predominantly that one type of stimuli, such as somatosensory association area, right? Or visual association area. That was still pertinent to just that one sense. But these multimodal association areas are broadly mapped across the brain. They're big regions, and many include mixes of sensory, motor, and association areas all converging together. The way I like to think of it is we perceive the world as one, right? It's not like we are first thinking about what we see in front of us, and then we switch modes and we hear things in our environment, and then we switch modes and then we smell things, right? We live and we perceive the world as one whole big unit, okay? And so we need those association areas to all converge together to give us that characteristics, right? I'll give you an example of speech. Speech is a complex action, right? So listening to speech involves primary auditory area. And then we transmit that information, remember, to Wernicke's area when it's language, or auditory association area if it's these sounds like woof, bang, pow, right? That'll go to the sound area, the auditory association. But if it's language, it's Wernicke's. Then, right, in multimodal association areas, such as maybe in the back over here, or perhaps in the prefrontal cortex up here, there's some multimodal association areas where signals are interpreted all together, right? So all of the senses come together and memories come in. Oh, yeah, I remember smelling that smell, hearing the voice of, um, I don't know, someone I knew, right? That the perfume they're wearing, whatever. And even emotions will go into that area, right? There's emotional areas of the brain in the limbic system that'll feed into this multimodal area. And even there, things get mixed, and you know, you tie all of those sensations to your emotions, and that gets stored as a memory, right? So here you see how it's a lot more complex than just our association regular areas, right? Multimodal means you bring it all together. Um, and so from speaking, right, if you hear someone talk, in those multimodal areas, you kind of perceive everything. You remember what words mean, right? You get your emotions, right? When if someone's screaming at you, then you'll be very thrown off. If someone's saying you something nice, you'll be flattered, right? Well, depending on that, right? Then those multimodal association areas, right? They will send signals to places like Broca's area, right? So that you can respond back. Okay. So I guess my point here is that, we talked about just afferents and sensory pathways going up, all the way to something association cortex, right? And then we talked about motor things starting in these motor association areas and then going down. But I never linked the two together, right? For example, here you hear someone say something, it goes to primary auditory area, then it goes to Wernicke's area, but then I need to link that somehow so that I could then respond back to them, right? So these neurons here in Wernicke's area, they'll actually go to the prefrontal cortex where these are multimodal areas, and your memories, emotions will remember the words and the feelings that you want to convey, right? And then that'll talk to Broca's area, right? And it'll tell it to say certain sentence, right? And then that'll go to primary motor area where you'll move your jaw, your tongue, and your lips, and then that'll go down to those muscles, right? I guess my point is we saw this in the sensory afferents, we saw this in the efferents, and now I'm just adding a piece in the middle. This multimodal association area is where the middle ground comes in, and we link sensations and motor, what we want to do next, together, right? The reason why we sense all these things, right, is because then we can do something about it, about those sensations. So this is a perfect area where we link things. And on top of it, memories and emotions are added. An example of a multimodal association area is the prefrontal cortex. It's found in both the frontal lobes, and it receives signals from many association areas. This whole area here, prefrontal cortex, right? We always left it blank here, but this is a multimodal association area, which is very interesting. One of my favorite parts of the brain. This forms your intellect, your personality, most importantly, motivation to do things, right? your consciousness, your ability to plan, reason, learn, problem solve. This frontal region here, right? Um, it really is the place where you can do this problem solving and have motivation, right? After you've sensed all these things in the world, right? And you've heard things, you've seen things, right? You've smelled things, it all comes together, and then you need a want to do something about those things, right? And that's your prefrontal cortex, this want to do, um, I don't know, once you smell the smell of a perfume and you see a person, right? Then you realize, oh, this is my friend, right? And I should hug them. I haven't seen them in a while. And this want to hug them will be in this prefrontal because you associate all these senses of seeing them, smelling them. Um, I hope they smell good. And then hearing them, right? And then you decide to go and move. So then you'll start in this area, you'll send a motor pattern out to go and run towards them to give them a hug. Um, so this is a very interesting brain region. Um, do you see how all these two parts of the brain are motor, right? Well, prefrontal cortex does have some motor abilities too. Like it doesn't make the plan. It starts even earlier. It gets the desire to do something, the motivation. So I like to think of the frontal lobe as the motor area of the brain. And when you start down here in the prefrontal cortex, this is very complicated motor area. This is like the desire and the motivation to do something. Should I get out of bed today to go to school, right? Then you send a signal to an association area, right? Maybe Broca's or this motor association area. And from there, right, you make a motor plan, we said, such as, okay, I'm gonna, once you've decided down here in the prefrontal that you want to go to school, right, then you send a neuron to this motor association area and say, okay, move your arm, move your leg, sorry, move, get yourself out of bed by grabbing the side rail and lifting yourself up, right? And then over here, you'd press all the buttons on the muscles of the body in primary motor area. Interestingly, patients that are suffering from depression, when you put them in a scanner, this prefrontal cortex is very dimly lit compared to the regular. It makes sense. This prefrontal place is where you have a lot of motivation. A lot of times, patients, when they're depressed, their personality will be changed a little bit. They're not going to be themselves. Maybe they'll be a bit more irritated, right? Because this area is not function as it usually does. Your ability to plan things, to problem solve, is diminished here, right? So all that to say, depressed patients, right? Oftentimes this region over here where that has these motivational areas, um, there's a reason why someone with depression has difficulty just with showering, right? Getting up out of their bed is a difficult thing to do. Brushing their teeth, their hair, right? All of these things are motor things to do that everybody, all of us that are not suffering with depression, we can do every day, right? Very easily. But if prefrontal cortex has diminished signals due to depression, then we can't really do the things that we want to, right? So there's a neurobiology to all of these symptoms that depressed patients have. Anyway, that's my tangent. I could go on. This prefrontal cortex has many connections with the limbic system, and it integrates a lot of sensory information and predicts consequences, like I was saying. A lot of those sensory infos all converge here, right? There's a few other areas that are multimodal association areas. In fact, the limbic area is a multimodal association area. Okay. Um, Limbic association areas is entirely linked with the limbic system to provide emotional impact and establish memories. So, all of these areas that are responsible for emotions. This is an interesting one. Posterior association area over here. This is responsible for generating a mental map of our body in space and creating a perception of the world. Um, how can I put it? This is really one of the first areas where all those sensory association areas converge. And as I was telling you, we perceive one world, right? We don't, like, there's not channels in our brain that switch from visual to hearing to smell, right? We see it all together, we hear it all together, we smell it all together. Our world is one. And that's because over here, all the neurons from this somatosensory association area converge here. The visual association area converge here. The auditory association areas converge here. And everything makes sense as one world. This is where we perceive our world all as one. Prefrontal cortex, as I told you before, that's a little bit more on the motor aspect of things, right? Actually, once we converge everything in posterior association area, often we'll send neurons over here to the prefrontal cortex. Because once we're in the prefrontal cortex, then we can start thinking about what we want to do and our motivations to do things. So this is more a converging of sensory info, and then we send it over here to these areas that are closer to motor abilities. All this to say, I want you to understand how information is carried between cortical areas. Um, in my head, there's three levels of complexity. There's the simple areas that are basic babyish, such as primary sensory areas. For example, primary somatosensory area is only carrying information about a neuron in one little patch of skin feeling one type of, I don't know, crude touch in that patch, and it sends that neuron all the way up to this primary area. This is very basic information about one tiny piece of the body. Then it gets associated together. That's level 2 of complexity. All of that sense, such as touch, right, all gets associated, and you end up realizing, oh, I feel touch across my whole left body. I must have a blanket on me, right? You associate all of those touch feelings all together. And then you associate all the senses together, right? So touch comes in, um, taste, right? Vision, hearing, all converge. All of those association areas converge onto a multimodal association area, right? Um, and then here they could really be interpreted all together as one big world and what's going on, right? And then if you decide, right, you could decide to do something about that perception of things, right? For example, you might hear someone insulting you. You might see someone who's angry, right? You might um, feel someone who's punching you, right? All of this will be associated in this multimodal association area, all the senses together, and you're going to want to fight back. So you're going to send a signal right to motor association area and say, punch them. This is getting violent. You're going to tell them to punch them. And then that's a motor plan in motor association area, right? And then you'll send that to the motor areas. You'll contract your bicep, you'll relax your tricep, you'll contract other muscles in your hand that'll allow you to punch them, right? So I guess now you see the whole picture pieced together. We saw this picture in one of the lectures, we saw this picture, but I guess multimodal association areas, they really converge the two sensations and motor ability together, right? All right. Um, what is memory? It's a system of storage and retrieval of information, right? So memory storage occurs in different stages, actually. Information in short-term memory, we call this STM, not the STM bus and metro, helps us commit small packets of information for immediate use, okay? I think mine is totally broken. Over here, you see that you have sensory input, right? Maybe someone is screaming out a phone number, right? And sensory memory, right? You try to you get these sensations, right? This is me at all times, forgetting almost immediately. If you pay attention to it, and we actually tell you, please, like, focus on this phone number, right? You will all probably be able to commit it to short-term memory, words, names, numbers, right? You can maintain this by rehearsal. Um, you could also forget it again. We have limited storage, right? Phone numbers are a good amount of numbers to remember, but if it was any longer, we wouldn't be able to, right? And then long-term memory, LTM, allows us to store almost limitless information, and we store this for um, long-term use. How do we transfer memories from one stage to another? This word is called consolidation. To consolidate something means to really bring it together and have those memories consolidated into your long term, right? They're brought together and they're kind of anchored into your long term. They're these little facts, right? These sensory inputs, they start in short term, but they could really get consolidated. And how do we consolidate memories? Emotional states help us to transfer long-term memories. I always tell you when you're studying, all the stupid little acronyms that I give you or the weird analogies, right? They're there so that maybe you try to laugh, or you're like, this teacher's crazy, and it's an emotional state that'll help you to consolidate it a little bit longer, right? Or colors help to do this, right? To remember that. If I give you just this PowerPoint in black and white, it might not help you consolidate those memories. Repetition helps to consolidate memories, okay? Um, you could repeat things over and over, and then it'll go to your long-term memory, right? Um, I don't even remember phone numbers anymore. What else do we have? And associations also help us to have this. So, when I tell you to study, I tell you to not memorize everything, but to associate concepts, right? This, see, it's biology. It's about memories. So this is how we can go from this shorter term to longer term. It's called consolidating. Where do memories occur? Certain areas of the brain are responsible for different types of memories. So we have two big groups of memories. We have declarative memories, which are factual memories. For example, sensory information goes through the normal cortical areas, after which this information is circuited between the thalamus, the limbic association areas, and the prefrontal cortex. These are factual memories, okay? So over here, these declarative memories, things you know that you can tell others, right? For example, it could, and there's many different divisions, but these episodic memories, something like remembering your first day of school. This is a fact, right? This is something that is these declarative memories with sensory info. Knowing the capital of France. This fun fact. Um, this is going to be a declarative memory, something you know that you could tell others. Non-declarative memories. These are memories of events and procedures. For procedural memories, sensory and motor signals are circuited to the basal nuclei, the thalamus, and the premotor cortex. So, non declarative memories or procedural memories, things that you know that you can show by doing. It's not something that's easy to describe, right? For example, knowing how to ride a bicycle, right? You to describe this, it's pretty tough, right? It's more of a procedural memory. When you're on the bike, your basal ganglia, your basal nuclei and your thalamus, that they're gonna know immediately what to do, right? Won't even need to think about it. It's hard to describe to someone how to do that. Being more likely to use a word you recently heard, right? Like a recall, right? This is like kind of stored there. Or salivating when you see a favorite food. This is this is conditioning. You're not really in control of this, right? So don't worry too much about this slide, but know that there's two types of memories. There's these declarative memories, things you could tell others. These are facts, right? um, and non-declarative things that you can only really show by doing it, right? It's it's kind of, I don't want to say involuntary, right? But like it's something that you've done through some kind of procedure, right? Um, Next thing, we're going to take a look at how we measure. I'm going to just skip that slide 2 seconds. I just want to show you how we measure brain activity. Um, and this is so funny because I just came back from a sleep test at the hospital, and I was hooked up to these crazy EEG electrodes. And so, brain activity, we have two ways of monitoring it. We could monitor blood flow throughout uh, kind of the brain and regions. So this is called functional magnetic resonance imaging, or fMRI. And as I said, this kind of, it's not measuring the electrical activity per se. It's measuring the blood flow, which is a proxy for what areas are lighting up and charged. Another way you can take a look at brain activity is via neuronal electrical activity with an electroencephalogram. This is not an ECG, this is an EEG. Um, Electro means you're measuring electricity. Encephalo, we know that anything cephalo means brain, right? And gram is to measure, right? We're measuring brain waves. Um, Action potentials, we know, they generate electrical fields, and brain waves are rhythmic patterns of action potentials produced by many neurons together in a certain region. And actually, there are four wave patterns that you could measure with an EEG. Um, when we are relaxed, right, which happens rarely, these alpha waves are going to be found. And how do we know they're alpha waves? They're 8 to 15 hertz. And so you see this frequency, right? Do you see how the waves are very spaced out, right? That's an alpha wave. Beta waves, 16 to 30 hertz, they're present when we're very focused. It looks like a focused wave here. You see how the time between the waves is very small, right? Because the frequency of the waves is a lot higher. So this is a higher um, frequency of waves. They're called beta waves. And so they're present when we're focused. Literally, if I put an EEG on your head and I asked you to focus on something or read something or think of something, you'd see these these waves over here. Whereas if I asked you, if I just put calm music, you would see these alpha waves. Your neurons are literally firing less, right? Remember, these are recordings of a group of neurons, right? Um, Theta waves over here are much slower. Do you see the amount of time between the waves? They're normally occurring in children, right? Because children are still not fully developed, but they could also be present during deep relaxation, right? So you could measure these 4 to 7 hertz. Look at the space between, much bigger space between than the alpha waves. Or these delta waves are zero point 0.1 to 3 hertz. Look at how much space is between. The frequency of the waves is very low. There's not much frequent waves. There's only a few waves. And so these are present during deep sleep, REM sleep, but they indicate brain damage if they're present when you're awake, because that means that you're not at this, theta or alpha level. Okay. So during my sleep test, they were probably measuring, hopefully, these REM sleep waves, right? They could tell when I was in my REM sleep based on the delta waves. All right. I just want to go back now that we talked about it. This is an EEG. They'll put little, oh darn, I should have put a picture of what my setup was. Over here, you have these little electrodes, right, that are on the head. They're not zapping you, by the way. They are just sitting on your scalp, and they're um, detecting the waves that are coming out of your brain. Right now, your brain is emitting waves, right? And then this looks scary, very scary, but this is not a needle. Inside this syringe is a gel that they insert in the little hole here so that the contact between your skin and the electrode is there's a good contact to measure the waves properly. So they insert jelly so that there's some gel all over the electrode. So I came home last night, sorry, this morning, and I had to wash my hair 5 times. I was full of this disgusting gel that they had to put. So this picture looks a lot worse than what it actually is. Again, these are just measuring waves coming off the brain. They're not electrocuting you. There's no zapping. They're just sitting on your scalp, picking up the waves. There's really nothing to it other than that. And this fMRI here, we didn't talk about fMRI that much, but that measures where blood is flowing in the brain, right? So it could image your brain very nicely. And so there's a big magnet that turns around and collects that a nice picture of your brain and where areas are lit. Okay. So, voila, we're good with that. I want to take a second to show you this slide because I think it's extremely interesting when talking about, mental health. Um, I think more and more it's becoming normalized that this is the same idea as having a physical health problem. But if that wasn't proof enough, right? Look at these pictures. You truly do see, I hope, in these pictures that for these different mental health conditions, the brain pattern is completely different, right? So over here, this PTSD patient, right? This is a normal brain when asked to do a specific task, and this is a one with PTSD, and there's a lot of areas of the brain that should not be lit up, right? Um, and so this, there's a neurobiology to this. Over here in the depressed brain, um, you see that this normal brain, right? You in the frontal lobe, this is the most important part, and the the sensory areas, right? They're all lit up, and then here you see the frontal lobe is dark, 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 right? And even these regions here that are lit up are probably some very emotional areas, right? That really are involved in the limbic system. Again, this slide, you don't need to know uh, anything about it. I just want to take a second to show you really the neurobiology behind mental health, right? Here you have bipolar disorder, and you have these differences in the two. These are types of brain imaging I didn't talk about, but they're very particular to a certain condition. Here you have a brain of an ADHD patient, right? And you see that actually there's not a focused of information in one specific region. So all that to say, you don't need to interpret these, but I think it's nice to see how brain imaging can help prove, not that we need any proof, but someone that has a mental health condition, really it does have some kind of neurobiological underpinning to it, right? When you break a bone, you go do an x-ray and you see that there's a difference from what it should be normally. Same situation with these mental health disorders. You see that there is an imaging difference between what it should normally look like, right? And I think people who haven't been through any of these, right, um, don't truly understand what is behind all of this, right? What could be there. Um, and, thankfully, we won't all have these four diseases at some point in our life, but I hope at least we can realize that there is something about it, right? That these brain patterns really do show that there is something that we should not let go, and just say things like, what happened to you wasn't even bad, right? Get over it. Lots of people have it worse. These are things that maybe, way back when were very prevalent to say, but now um, we understand mental health a lot better. The next part I need to show you is about lateralization. Um, this is the tendency of some unique neuronal functions to occur in one hemisphere but not the other. How do hemispheres differ? Physically, they're almost identical. They perform many of the same functions. Most of the functions are mimicked and mirrored on both sides, right? And actually, the things that we do on the right side of our body move on our right side, they're processed in the left hemisphere. The things that we do on our left side of our body are processed in the right hemisphere. So, um, you know, uh, they're very similar, but um, you know, they perform many of the same functions. However, certain tasks are coordinated by just a dominant hemisphere. Sometimes there's some things, and I'm going to show you things like language, that is just on one hemisphere. Um, what is cerebral dominance? It is a property wherein one hemisphere dominates a task. For example, most people, right, 90% of the population is right-handed, meaning that their left hemisphere is the dominant one, right? Remember, things always cross to the other side. So 90% of us have a left hemisphere that's dominant because we are right-handed. The rest of you, 10% people, you are right hemisphere dominant, okay? Um, that is, this hemisphere is primarily responsible for things like language, math, and logic. These are three things that really only are on one of the hemispheres. So, hemispheres don't operate independently, actually. Communication across the corpus callosum is pretty instant. There's connections between the two hemispheres. Cortical areas are similarly arranged on both hemispheres, but the multimodal association areas on both hemispheres, they're differently activated to accomplish an overall task. I think the point here, what I want to say is be careful. You see in the, I don't know, people have this idea that we're left brain or right brained people, right? Oh, if you're more creative, logic and you're on this hemisphere, right? But logic and creative personalities do not correlate with cerebral dominance. Most people are left brain dominant, 90% of the population actually, right? Since we're right-handed, and it doesn't mean that their personality is more creative, right? So, what types of tasks are coordinated by each hemisphere? Tasks of the dominant hemisphere. So, for most of us that are right-handed, right? Um, these are the types of things we'll find on our left hemisphere, right? Remember, things cross. So, the dominant hemisphere perceives the boundaries of the body and is most responsible for analytical tasks, right? Linear thinking, math, logic, time perception. Broca's area and Wernicke's area are only on the left side, actually, interestingly. Um, and creating rules and establishing an ego, actually, these things are also located on this left hemisphere if you are right-handed. Now, I also want you to remember that this left hemisphere is lateralized, meaning, you know, the opposite side of the body, including sound, vision, touch, is all information from that opposite side, right? So all of seeing on this side and feeling and hearing goes to the left hemisphere. And seeing and touching on this side goes to the right hemisphere, right? Um, the non-dominant hemisphere, so for my right-handed people, right, most of us, we have our non-dominant hemisphere on the right side. So what do we have here that's different? Facial recognition is here. Um, the big picture thinking, right? Emotional contexts, empathy during our communication, right? Intuition, spontaneity, artistry, all of this. This is where the idea of the creative mind kind of comes in, but it's not quite accurate, right? There's not People aren't more creative because of that hemisphere, because we know that we have the corpus callosum that can talk to both sides, right? Um, and additionally, I want you to keep remembering that we're still controlling the opposite side of the body. For everything else, though, right? Like touch and vision and hearing, all the information from the left side is going on to the right hemisphere, right? Okay, so now we're going to move on to lecture 9. We wrapped up that other lecture. This is quite a quick lecture. We'll take a look at cerebral spinal fluid, how it circulates, and then we have a little section on reflexes. If you see this brain here, right, what is being shown are the ventricles of the brain. We're going to take a look at those. So you have your key takeaways, your objectives, you have additional resources. I put some very good videos here about how CSF flows because it's very hard to show this in 2 dimensions. So first, what are the chambers of the brain and the spinal cord? In the CNS, we have chambers. These are spaces connected to each other so that CSF, cerebrospinal fluid, can circulate and continuously flow in a unidirectional manner. This is a one-way avenue. Over here, you see these big C rings on each side, right? There's one over here and one over here, these big C rings. These are the lateral ventricles. Look at the brain. They're more lateral, not medial, right? they're to the side. So these are the two lateral ventricles. We also call them ventricles 1 and 2. So ventricle 1 and 2, you don't need to know which side they are. We actually, I don't think there is a clear logic to that. And so all of these, I want you to imagine this big C ring filled with CSF. Okay. Then CSF goes through this little hole here, and it goes into this third ventricle, which is a smaller space, but it still is a space. So the third ventricle, CSF flows in this area, right? Then that CSF will move down this little canal here, down here, and go into ventricle number 4 near the cerebellum. And then ventricle 4, right, is actually connected, if you follow it through, to the central canal of the spinal cord. Do you see this middle part here? That's the little hole that you find in the spinal cord, that tiny little hole in the middle, the central canal, that's linked to ventricle 4, and it goes all the way down the spinal cord, right? So the chambers in the CNS, right? We have 4 ventricles in the brain, 1, 2, 3, 4. 4 spaces, 4 chambers, right? And we also, in the spinal cord, we have what we call the central canal that goes down the middle, okay? Ventricles 1 and 2, they merge together, right? And connect to ventricle 3 via the interventricular foramen. Inter means between the ventricles, right? Foramen means a hole, right? So do you see this little passage here? It allows the CSF in ventricle 1 over here to go into ventricle 3. Ventricle 2, it goes into ventricle 3, right? And so all the CSF merges into this one chamber, right? Then ventricle 3 connects to ventricle 4 by flowing into what we call the cerebral aqueduct. So connecting ventricle 3 and 4, this aqueduct here, that's called the cerebral aqueduct. An aqueduct carries aqua, water, so it makes a bit of sense. It's carrying CSF down to the 4th ventricle. Here you see it in this picture right over here, connects to the 4th ventricle. Now, the circulation of CSF. We talked about what the chambers were, what the passages were. First of all, what is the function of CSF? Number 1, it's very good for physical protection. This includes the fact that it bathes both organs, the brain and spinal cord, and cushions them from absorbing any trauma or shock. I believe, if I threw you off the 3rd floor of AME, you would much rather want to hit a waterbed that was at the bottom than hit the cement asphalt, right? That's because water, it takes the hit of the impact, right? It's kind of like an airbag, but that's air, not water. And it prevents, it absorbs a lot of the shock, right? So let's say the brain, if it's not too bad and it's not concussion, then the brain will kind of bounce in this liquid, right? And the liquid will absorb the shock instead. But the main goal of the CSF, right, is chemical protection. So the brain is bougie, remember. It does not directly contact the blood, and instead it uses CSF to distribute nutrients, gases, wastes, and hormones to and from the CNS. So instead of using blood, the brain and spinal cord uses CSF to feed the inner parts of the brain and the outer parts, right? It uses this bougie filtered blood called CSF. Over here, I want to take a look at this picture. First of all, let's remind ourselves of what our layers are, right? Do you see in gray, we have the dura mater, the outer dura, and the inner dura, right? Here you see the dura of the spinal cord in gray as well. In pink, you see the arachnoid matter that's on the middle layer. And the pia matter, actually, you don't see it, but it's stuck on the brain and on the spinal cord. You don't see this pia matter. It's really on the brain, and you saw this in the brain when you did the dissection, right? All the pia was in the nooks and crannies of the brain. So we have, remember, it's called pad, pia is on the inside, arachnoid is in the middle, and the dura is on the outside. Do you see how for the brain, though, the dura has two layers, the inner and outer, and it makes this sinus over here, right? We'll talk about the dural sinus soon. Now, I want you to know that bathes and exposes, CSF bathes exposed CNS surfaces. So the CSF bathes the brain and spinal cord inside the ventricles. Do you see the ventricles here? We have, if I cut the brain in two, right? Remember our ventricles from here. If I cut the brain in two, this would be my lateral ventricle. This would be my interventricular foramen. This would be my third ventricle flowing down through the cerebral aqueduct into the fourth ventricle, into the central canal, right? So all of the ventricles of the brain inside, the CSF feeds the neurons that are around these ventricles and around the central canal. All this spinal cord needs to be fed with nutrients, with oxygen, right? Also, the outer surface of the brain and spinal cord are also fed. This is called the subarachnoid space, right? Under the arachnoid, between the arachnoid and the pia mater, is this space here. Do you see this space all around, right? This over here in green, I've labeled it. You see the subarachnoid space of the spinal cord. You also see the subarachnoid space all around here, right, of the brain. This is below the arachnoid matter, and sandwich in between the pia is like touching the brain, right? So this is the subarachnoid space. So I want you to know, we're going to take a look at where CSF flows in a minute, but this is where CSF goes all over the brain, on the inside, the outside, all over the spinal cord, on the inside, in this little canal, and the outside, right? How is CSF made? Blood plasma passes through and is filtered by the choroid plexus to produce CSF. What is this choroid plexus? So, the blood cerebrospinal fluid barrier, it occurs in the choroid plexus. Basically, as neurons are going towards the brain, right, all as they're going along, remember that these blood vessels, all these blood vessels going towards the brain, they are, do you see these yellow little astrocyte feet that are hiding the blood vessel, right? They're wrapping around the blood vessel. That's the blood-brain barrier. Everywhere in the brain, there's this blood-brain barrier blocking blood vessels, and so nutrients cannot get to the neurons. These neurons cannot receive the nutrients from the blood-brain barrier. But when they go and they reach this choroid plexus, okay, then this choroid plexus, right, is an organization of capillaries lined by these ependymal cells, right? These ependymal neuroglia that have tight junctions between, so they don't let much through, but they do have ion pumps that will modify the composition of the filtrate. So blood passes, the blood vessels, they're all wrapped by these astrocytes until they get to these special regions called choroid plexus, right? A plexus is a tangling of things, of blood vessels in this case, that are lined with the ependymal cells. And the ependymal cells, they have little pumps, and they could put into the CSF a bunch of things. ions. They could put in glucose, most importantly, right? All of these things will go through into the CSF, and all of the unwanted things, right? For example, viruses that could infect the brain, right? Or different drugs will go and continue along and not get put into the CSF, right? So that's the choroid plexus that makes it. Where is the choroid plexus? It's located in all four ventricles of the brain. Do you see this purple fuzzy stuff? It's trying to show the choroid plexus in the ventricles. So on top here, in the lateral ventricle, you see the choroid plexus, and there's one in ventricle 1, and you don't see ventricle 2 here, but there would be one in each ventricle. On the bottom here, right after the interventricular foramen, you have ventricle 3, and you have some choroid plexus over here, right? And then over here in ventricle 4, you have a little bit more choroid plexus. So in all four brain ventricles, you have choroid plexus, okay? The four choroid plexi are lined with the ependymal cells, right? All these choroid plexi have ependymal cells, and they filter the plasma, right? When the blood comes near, they can filter the plasma and make CSF that's full of nutrients, full of good stuff, oxygen, right? And all of that liquid will go in here and it'll fill the ventricles up, right? You see all this blue spot? That will be due to the production of CSF from these purple little choroid plexi that make CSF in this space, in the chambers, okay? So let's go through the steps. I need you to know very well where CSF circulates. Step 1, the choroid plexus of ventricle 1 and 2, right over here. These are the lateral ventricles, right? They make nutrient-rich CSF. So these are the producers of CSF, and they put all their CSF into this lateral ventricle here, okay? Then CSF flows down via the interventricular foramen, that's this, between the two ventricles, interventricular foramen to ventricle 3. In ventricle 3, this choroid plexus of the ventricle makes and adds more nutrient-rich CSF, right? So it adds to the CSF that was made from up here. It's just pouring more liquid. More of this CSF is adding into this chamber, right? CSF then flows down, right? It goes down the cerebral aqueduct here into ventricle 4. The choroid plexus right here of ventricle 4 adds even more nutrient-rich CSF, right? Step 6, CSF can take one of two routes. Either this from ventricle 4, CSF can flow through the central canal here of the spinal cord, or CSF can flow through these apertures, these little holes on the side, into the subarachnoid space. Do you see this blue space here? That's the outer layer, right? That's the outer space in between the meninges, in between the pia, which is touching the brain, and the arachnoid, which is in pink, right? So it could flow in this subarachnoid space all around, right? All this subarachnoid space here. Do you see this subarachnoid space here? To other parts of the brain and spinal cord, okay? Do you see how also here it goes in the subarachnoid space of the spinal cord, right? Part 8, these arrows eventually, this is unidirectional, right? Eventually these arrows, they come up and, all these neurons that took, all these neurons are taking the good nutrients, the glucose, and taking all of the oxygen and the ions that are needed, but they're throwing out all their garbage, right? All of their waste. So this nutrient poor CSF right, that got all of its nutrients taken away from the neurons that were greedy, it eventually reaches these arachnoid granulations that we'll talk about in a minute, and they drain into this big dural sinus here to mix with the blood. And then it goes down to the blood system, and actually you don't see where it goes after this, but it goes down to the heart. So let's talk about this drainage because it's a little bit difficult to understand. Neurons, why must we drain CSF, right? Neurons absorb oxygen and nutrients from the CSF, and they release CO2 and waste into the CSF, right? Throughout this whole pathway, right? As I told you that CSF is flowing. What was the whole point of that? Is that this tissue here needs some oxygen and needs some of that glucose, right? This tissue here, this brain region needs some of that oxygen and needs some of that glucose from here, right? So we're going around feeding all of these neurons. They're throwing their waste and CO2, right? But then eventually, the used up CSF, the nutrient poor CSF, must now return to the bloodstream, right? We need to bring it back to the heart to reoxygenate it. So where does CSF exit? Over here, you have the pia mater. You have the subarachnoid space over here, where the spider webs are. And you have this arachnoid matter, right? Nutrient poor CSF in the subarachnoid space, right? Remember, the last step here that we saw was we're in this subarachnoid space going all the way back, right? Subarachnoid space here going all the way back. They move to arachnoid granulations. These, if you see at the top over here, you see these bumps here? These little grainy bumps? They look like little broccolis, right? Nutrient poor CSF in the subarachnoid space move to arachnoid granulations, which are small grainy projections of the arachnoid matter, right? They project out into this dural sinus. So nutrient poor CSF, right, crosses all this nutrient poor CSF. You see here, there's all this CSF full of waste in this area. It will go through the arachnoid granulation into the big tube here called the dural sinus. So all this CSF here goes through the arachnoid granulation into the dural sinus, right? Into this big tube here, this big dural sinus, which mixes with venous blood. That's why it's blue. It's deoxygenated. It mixes with some blood here. Do you see this big vein over here? That's the dural sinus, okay? It is directed to the back of the skull. So all of this goes and flows down to this area here. And that's called the confluence of sinuses. I don't care about that. But it's a converging of the sinuses. And from here, CSF and blood, right? This is a mix of now all the CSF and blood, which is practically the same thing as we talked about. All of this goes into this area, goes down through the jugular vein, where it mixes with more blood from other regions of the head, right? The neck. And this is transported to the heart, the lungs, the kidneys, and it'll be reoxygenated, refiltered, and it'll return back up to the brain afterwards. Okay? So, based on what you know of CSF composition, what would red blood cells in the CSF indicate? Well, it would indicate that the blood-brain barrier is not working properly. If I go back over here, red blood cells should not be entering into the CSF. So there's some kind of breach in the blood-brain barrier. And a hemorrhage would not be good, right? What could cause hemorrhaging? If we break the meninges, right, we could have some kind of hemorrhaging happen. That wouldn't be good. What problems would occur if there was a blockage of CSF flow, right? What happens, right, if For example, over here, the medulla starts swelling up and compresses this little canal, I don't know, right over actually the pons here. If they start compressing this canal, what would happen, right? Actually, there would be a backlog of CSF. You know this, when a pipe gets clogged here, it would backlog here and the lateral ventricles would expand, expand, expand and get big, big, big. The lateral and the third ventricle, right? And this would cause, right, a big pressure to accumulate in the ventricles. All of this area here, these areas would get expand, expand, and this is called hydrocephalus, right? When you have this accumulation of pressure, hydro means like water or pressure, right? Cephalus of the brain, right? When you have this expansion, that's not good. You're expanding the brain, and hydrocephalus has different symptoms in children and adults, right? Why is that? In children, you might see this situation. Remember that the bones, they're still not fully fused together, and they could actually cause it looks like the brain is bulging, right? But it's actually just the liquid that's there, right? That's expanding those because the bones aren't fused yet. In adults, though, what happens, those bones are fused, and when you have an accumulation of this CSF and it's expanding the ventricles, right, because of a blockage, in humans, that will press down against the brain and it'll cause a really big damage to the brain if it keeps accumulating. You see this in the shows, and they actually do this in real. When there is this symptom, right? This pressure in the brain. First of all, your body goes crazy. You start, your whole body overreacts, and you start fainting, and you start, probably convulsing and all these things. They're actually gonna go and drill a hole into the skull. They're gonna pierce the meninges to allow that pressure to be relieved at the cost of, it's not that bad actually, because you're going to be relieving that pressure, and all of a sudden that pressure won't be creating harm on the brain, right? You'd much rather, pierce through the entire skull and break the meninges and allow that pressure to be removed, right? Than to actually have it be pushing on a brain structure and injure the neurons forever, which can't duplicate and make copies of themselves, right? Okay, last thing, guys. What are reflexes? Um, a reflex arc is an evolutionary adaptation to deal with a sudden change in external or internal environments. Some stimuli cause the body to withdraw from a certain thing, such as a painful stimulus, or sometimes it'll allow you to move into a stimulus, such as a bladder contraction that keeps going and going and going to tell you to go pee. So, what are reflexes? They're rapid, they're autonomic, right? They happen automatically. They're prompted by very specific stimuli. Think of the knee patellar one, right? It's you hit right in that region and it's prompted by that specific stimuli. They show little variability. On a Monday, your leg won't kick to the right, right? It'll always keep kicking straight. On Tuesday, it won't kick to the left, right? It'll always keep kicking straight with the patellar reflex. And it helps preserve homeostasis. We're going to see how this is done. Transmission of information during a reflex. During any reflex, most information integration occurs in the CNS. A reflex could be a cranial reflex in the brain, or it could be a spinal reflex, right? Reflexes occur actually in both regions. This is kind of like a cranial nerve or a spinal nerve, right? We're going to see that in a minute. A reflex is not the only response to a stimulus, though, right? But it is the fastest one that you'll first see. For example, when you touch a hot surface, right? You reflexively withdraw your hand And this travels quickly. You see this blue neuron, it travels quickly up, 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 right, to this region. And it's carried to the CNS, and then it goes onto an interneuron. It goes onto this motor neuron, right? And then it goes and tells your muscle to quickly contract away, right? When you touch something hot, you don't even realize it, but you pull back. This is called the withdrawal reflex. And why is it a reflex? There's three neurons in this path. One, two, three. That's so little neurons, right? So the fewer number of neurons, the quicker the signal is sent, the less likely you'll be in harm's way. You could move quickly. This is good evolutionarily. Now, after you've withdrawn your hand, though, you know that you will perceive pain from the candle that you touched. Your limbic system will cause you to be angry, maybe sad, you'll cry. The sympathetic system kicks in, your heart rate starts beating, right? If it really hurt. So you know that it must go up to the cortex if you're able to perceive these things, the limbic system, right? So it's not, the reflex is these three neurons, but actually there's a branch that you don't see over here that goes up to the brain, but it's much slower. You first withdraw your hand, and then slowly with time you'll kind of feel this pain, right? Reflexes, they could be characterized based on four things. First of all, the nature of the response, right? You could have somatic reflexes or visceral ones. A somatic reflex is a mechanism that automates control of your skeletal muscle. This is weird. Remember I told you skeletal muscle, I kept repeating this to you guys, it's always voluntary control. Well, I'm screwing you over now. It's always voluntary control 99% of the time, except for reflexes. So if it's a somatic reflex, somatic reflex receptors exist on the skin and in tendons, in muscles and ligaments, and they're going to generate indelicate and imprecise movements. So the little kick of your foot, right? This is a somatic reflex. These quadricep muscles here, you're usually in control of. But all of a sudden, right, you're not in control of it when the patellar is hit. There's also visceral reflexes. These are mechanisms that control the functioning of organs, glands, tissues, smooth and cardiac muscle. These are all things that are usually involuntary, so that makes sense. So visceral sensory information is interpreted locally or by the hypothalamus. For example, your cardiac, your respiratory cycles, peristalsis, these are all visceral reflexes, right? You're not in control of it. You feel stretch in your stomach, you send it up to the spinal cord, it comes out, and then it tells your stomach to contract, right? Reflexes could be classified according to their processing site. As I told you, if we process things in the spinal cord, right, then we're actually going to be we're communicating between sensory and motor pathways via spinal nerves. For example, this is the bladder reflex, right? To when you need to pee, you're gonna send stretch from your bladder. Nerve impulse will go up. Over here, it'll synapse onto this green interneuron, and then it'll go back down and tell the bladder to contract, right? Once it feels the stretch, it tells it to contract so that you could pee out. The cranial reflexes, right? This is an example of a spinal one because you saw the neuron just enter the spinal cord, came out. In a cranial reflex, communication between the sensory and motor pathways occurs in the brain via cranial nerves. The eyes, for instance, when you see the eye and your pupil dilates or you move your eye, right? This light goes in, it goes into this area of the brain, right, in the brain stem, and then it sends motor outputs out, and it controls the over here you see it's controlling the size of the pupil. Okay. The third way to categorize a reflex is by how complex it is. If it's a monosynaptic reflex, the sensory neuron synapses directly onto the motor neuron, which then synapses onto the effector. Here, the patellar one is an example of a monosynaptic one. You see this one neuron go in, it synapses onto the motor neuron directly and out. The less the number of neurons, the faster the signal. This is a very fast signal that we get, right? There's not even an interneuron here. That's monosynaptic. The motor neuron acts as the processing center and the effector pathway with minimal delay in signal transmission. Only stretch reflexes like the knee jerk are monosynaptic. Most are polysynaptic and they have interneuron, one interneuron or two that are used to relay the signal to the motor neuron. Interneurons process the information, which create a longer delay in the signal. Blah, 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 whatever. Here you see with the one neuron, it goes onto an interneuron and then out onto the motor neuron. The last thing is going to be how we classify reflexes based on when they develop. Innate reflexes. Innate means that it came from when you were born. It's internal. It's inner inside you since you were born. These are genetically programmed as their connections are formed during development. They're predictable reflexes such as pain responses, chewing, sucking, optic tracking of an object. These are all there when you're born. Okay? Acquired reflexes are generally more complex and they're learned and enhanced by repeating over and over and over again. For example, they're still rapid and automatic, but they're specific to the individual, how much you learned it, right? I'm going to say first walking, right? When you walk, you have this pattern and you learned that. You acquired that reflex, right? And eventually you don't even think about it because your cerebellum and your spinal cord have this pattern. Driving, this is the scary one. You know this feeling when you're driving and all of a sudden you kind of wake up from a daze and you say, whoa, where I just fell asleep for 20 minutes. It feels like I fell asleep for 20 minutes, but you were just in autopilot mode driving, right? That becomes a reflex at one point. Skiing becomes a reflex. You know the moves that you're making, or biking, right? When you first learn to ride a bike, it ain't so pretty and elegant. Consider the following. Both reflex types can be enhanced through repetition, right? Both of these you could, keep practicing, making sure that you know. Both can be modified to improve reaction times, and both can be suppressed actually consciously, right? You could actually go and not and suppress them consciously, um, but they can be distinguished based on anatomy, right? You can't really say, oh, this one is this or that. So the last thing I need to do is explain what the reflex arc is. The reason why it's called a reflex arc, it looks like an arc. Like it just goes in and out. There's no up to the complicated cerebral regions, right? Arrival of the stimulus, you step on this, you put your hand on this tack. The receptors detect the pain. They activate the sensory neuron. Sensory neuron goes onto this interneuron, which talks, gets processed. Then this motor neuron exits and activates the effector, and then you have a response by the peripheral effector to move your hand away from the harmful stimulus, right? When you touch a pin, you quickly retract. This is a withdrawal reflex example, right? So you could take a look at this on your own. This is just the steps of the reflex. So a sensory receptor is stimulated. The signal travels along the sensory neuron. The signal is processed in the spinal cord. A motor neuron is activated. The effector affects its change. Now I'm just going to give you the 4 examples of certain types of reflexes, and then we will call it a day. So here you have visceral spinal reflex. We're going to see the example of the urinary reflex. Visceral, right? We're going to see, so first of all, this visceral spinal reflex, the urinary one, it's innate, right? You're born with it. It's spinal because it's the bladder is closer to the spine, not the brain, so it'll go in the spine. And it's visceral because it's these visceral organs, the bladder, that's being controlled. It's polysynaptic, meaning it has more than one neuron, and it's ipsilateral. It's on the same side, so the muscle's on the same side. What's the purpose of this reflex? Is to inform you of your bladder fill level. The mechanism, sensory signals will cause the bladder contractions, which will increase in frequency and intensity as the bladder becomes more filled. So the sensory neurons, they pick up that, hey, I feel like over here you see in blue, it sends it to the spinal cord and says, hey, I'm stretched this much. Oh, sorry, that's in green. I'm stretched this much. In blue, you'll actually contract and then you could empty out your bladder because it says that it's full, right? Now, there's a caveat to that. We're going to take a look at it. So, again, the urinary reflex is that stretch receptors detect the filling of this bladder, right? The bladder wall stretching. The sensory neurons carry the signal to the spinal cord. The motor neuron is stimulated and it synapses with the smooth muscle on the bladder and causes the bladder to contract. But you know this very well, thank goodness. Contractions could be ignored by the thalamus, right? Your brain stem up here. You could send down a signal, right? And you could go and actually keep these sphincters closed, right? And you could say, nope, I'm not going to think about this right now, right? So you could concentrate on other things. And in order to pee, you need to open up voluntarily. Do you see how this is from the brainstem? You voluntarily need to open them up. So this is the reflex, the visceral spinal reflex, the urinary reflex. But this over here, right, is your conscious control of that. This is not a reflex. This is from the brain down. Let's look at an example of a visceral cranial reflex, right? So this is the pupillary light reflex. This is innate at birth. It's cranial, since eyes go through cranial nerves. Visceral, it's polysynaptic, and it's weird. It's ipsilateral and contralateral. Ipsi means the same, lateral, the same side. So you stimulate one eye and it comes back and affects the same eye. But it's also contralateral. You affect one eye and a neuron will also go to the other eye and have an effect. So the purpose of the reflex is to regulate the pupil diameter according to light intensity. So the mechanism of the reflex, when shining a bright light into the eye, the pupil will constrict, and in darkness, the pupil actually dilates. It's important. Shining light in one eye will cause the reflex in both eyes, since it's ipsilateral and contralateral. So what happens here? The optic nerve, cranial nerve 2 over here, picks up the light information and transports it to the geniculate nucleus in the thalamus. So it goes to the thalamus. We don't need to know the specific nuclei in the thalamus. And the signal is sent to a nuclei in the midbrain, which transmits the message to the motor neuron, the oculomotor nerve 3, and it'll send info to both eyes, right? And it'll tell the pupil to constrict. So again, this is visceral and cranial, right? It's a cranial one. It's the actual eye itself, the pupil size. It's not a somatic muscle. Somatic cranial reflex. This example, so it's somatic muscle, and it's cranial. It goes to the upper part of the body, the neck. The corneal blinking reflex, right? This is innate, it's cranial, it's somatic, it's polysynaptic, and it's ipsilateral. It only occurs on the same side. The purpose of this reflex is to protect the eye from physical and chemical irritants and excessive bright lights. So, sensory receptors on the surface of the cornea, over here on the surface of the eye, could detect irritants. Here they're sticking a freaking Q-tip in the eye. And that'll cause the muscles near the eye to blink and close your eyes to protect it, right? For physical and chemical irritants, somatic sensory signals on the cornea pick up signals which are transmitted along the trigeminal, right? That's what picks up the information. Over here, the touch here, you're going to go up the trigeminal nerve to the pons. The signal is sent to motor neurons in the pons, right? It goes on to these motor neurons here, along the facial nerve, right? Here's your facial nerve that's coming out, going to this somatic muscle, and to so you're exiting this facial nerve over here, right? And you're going to go to this muscle here and this muscle here to squeeze your eyes shut. You don't need to know the name of the muscle, just understand, these are somatic muscles you're usually in control of. You could blink your eyes, but it's a reflex in this case, right? When there's an irritant that's there, and it's a cranial reflex because it's going through cranial nerves. And lastly, we have somatic spinal reflexes. These are very common. These are the stretch reflexes, right? So for example, the stretch reflex, also called the knee jerk one, this is innate from birth, spinal, it's somatic. Usually these muscles are under your control, monosynaptic actually, and it's ipsilateral. Only this knee on the side that you're hitting will be affected, right? The purpose of the reflex is to regulate muscle length by sensing the degree of stretch of a muscle. So it's important in contracting postural muscles. So the patellar reflex, over here you see sensory structures called muscle spindles, which are on the ends of muscles over here. You see these muscle spindles, they're specialized skeletal muscle fibers that detect increases in muscle length. They actually detect how lengthened the muscle is. The stimulus in a stretch reflex is the muscle stretch, right? When the muscle stretches. Then it sends an information to another neuron, the motor neuron here, right, which triggers the muscle to contract to counter the initial stimulus. So first you stretched it a lot here, and then this neuron is actually hitting it back so that you contract the muscle. It's kind of detecting that the hammer is kind of mimicking as if it got stretched too much, right? And then this is trying to contract back, and it kicks your leg up to counter that initial stimulus. And the whole point of this is to prevent hyperextension. You want to prevent this whole muscle from overexerting and stretching too much. It's a balance to readjust the posture, right? Another type of reflex that's somatic and spinal is the withdrawal reflex. We talked about this, right? This one over here is spinal and somatic still. It's used to move away from a stimulus that's usually painful, temperature, or pressure sensitive. And so over here, you see this hand, right? Touches a hot object, you pull it away. That's because the pain receptors send a neuron up, the green one. It goes onto an interneuron, onto this other blue motor neuron, to the muscle to contract it to get away from the hot stimulus. Now, if you go to the next slide, you don't see it very well in this one. There is reciprocal inhibition because it causes the contraction of flexor muscles and it relaxes extensor muscles. So it excites one set of muscles and it inhibits the other. Look here. when you touch this hot stove, it goes in, right? And actually, this one neuron goes to the biceps muscle and it contracts it. If I contract this muscle, then I pull my hand away, right? But at the same time that you contract this muscle, you need to relax the antagonist muscle. So over here below is the triceps brachii, right? And you actually need to relax this muscle so that it doesn't overstretch too. So when you usually flex your muscle, your bicep will contract and your tricep gets relaxed, right? So this is called reciprocal inhibition. That these two muscles, the reflex actually goes to both muscles to tell them to do different things. Last few, the cross extensor reflex. The purpose of this is to move away from the stimulus and simultaneously support your movement, right? So this causes contraction of the flexor muscle and relaxation of the extensor. So um, you're activating on muscles on both sides of the body. Over here, you step on a painful object, right? When you step on this painful object over here, um, this reflex is more relevant with the weight-bearing limbs, right? So over here, this is what you stepped on. You want to remove your weight from this side, right? So over here, you see the typical, we saw it in the previous one, the withdrawal reflex is over here, right? But on the other side, you actually want to position yourself so that you could bear the weight of lifting up this leg, right? So the other muscle groups on the opposite side of the body, they're going to be signaled simultaneously, right? And they're going to be told to do the opposite of what's happening on the painful side. This is the withdrawal. This will be the pushing down reflex, the cross extensor, because it's the cross, the other limb, that's going to try to balance and keep that muscle stable, right? So that you could bear the weight of the body on it. And lastly, we have two more. The Golgi tendon reflex. The purpose of this one is to relax muscles if there's too much tension being created. Receptors structures called the Golgi tendon organ monitor muscle tension actually in the muscles, right? So the response includes relaxation of skeletal muscle to prevent excessive tension in that area. So let's take a look at it here. Tension activates the Golgi tendon organ. Here you have the Golgi tendon organ. It's like a proprioceptor, right? Inhibitory signals are sent to the motor neurons. Over here, you see that you have a sensory. You have this interneuron, and then you have your motor neurons, and this results in tension being reduced before the muscle is torn, right? So before we rip the muscle, then we're actually going to reduce the tension. And then last is the Babinski reflex, the plantar reflex. This is a reflex test that helps determine damage to nerves in the corticospinal tract. And we do this by stimulating the underside of the foot. So if you rub over here and you see the toes fan like this is called a positive reaction. If you do this rub over here and you see the toes go inwards, this is called a negative reaction. And the response depends on the patient's age. If the child shows a positive reaction, this is because their neurons aren't all myelinated yet, and they don't control their reflex very well. That's normal. In adults, what you expect is a negative reaction. You want to see the toes going downwards. That's the normal one. If you see a positive reaction in adulthood, that suggests neuronal damage to the corticospinal tract. And so it's a good way to determine that reflex. And that will be it. These two answers over here, they're both C. So you have the answers, take a read at them, and that's it for that lecture. We are done.